What are histone messengers and what are the genes that are involved in the formation of histone proteins? Now we know what histones are. Histones are the component proteins that form a nucleosome and help in the bundling up of the entire DNA structure altogether. Now, the question here is what leads to the formation of the histone proteins? What gene codes for it? So let us look at this at a more molecular basis. There are two types of genes that actually code for histones. The first type of gene is known as a dependent gene or the replication dependent gene. Then we have the independent gene, the replication independent gene. Both of these genes have one thing in common, that they don't have an intron in them. Both of them are free of introns. And the other thing that we can notice that the replication dependent gene is much more smaller and it has a small transcript and it has a small 3' UTR. The replication independent gene, however, is larger in dimension and has large poly A tails. But in this lecture, we're going to be dealing with replication independent gene, mostly with replication independent gene. So, replication independent gene. Replication independent gene is better understood if we look at the structure of a nucleosome. And so a typical nucleosome looks like this, a dissected nucleosome. Nucleosome is made up of histone proteins. We have the H2A, H2B, H3, and H4 that form the core protein. Now, what is the exact role of replication independent gene here? Replication independent genes are responsible for producing variants of the histones. So all of these histones have canonical forms. So what are these canonical forms? For example, the canonical form for H2A is known as H2AX and H2AZ. Then the canonical form for H3 is H3.3 and H3CID. Then there's also a canonical form for the histone protein that is associated with the linker or the H1 linker protein. The canonical form for H1 linker comes in form of H10 and H1T. Now, where are these genes? Where are these replication independent genes found? In the chromosome and in the human genome, where are these genes found? So, if you look at the entire genome, you can see that they present themselves in the form of clusters. So there are clusters in which there is a high concentration of these genes, independent genes, replication independent genes. There are three basic types of clusters. We have the HISD1, that's a large cluster. Then we have a medium HISD2 cluster, and then an associated HISD3 cluster that's much smaller in size. If you look at a chromosome, you can see that the large cluster is located, or HIST1 is located on chromosome 6, whereas the HIST2 and the HIST2 and the HIST3, which are the medium ones, are located on chromosome 1. Okay, so let us learn about the maturation of the histone mRNA. Maturation of the histone mRNA requires three basic steps. The first step involves the formation of the 3' UTR or the 3' untranslated region. The second step involves the translation of the mRNA by EIF4E or elongation eukaryotic initiation factor 4E, and then the degradation of the entire histone mRNA. The components that are going to be required later for this entire process that we're going to be looking at later are the stem loop binding protein, or the SLBP. Then we have the snRNA, or small nuclear RNA U7. Small nuclear RNA is about 57 to 70 nucleotides long and is mostly transcribed by the RNA polymerase II. Then we have HDE. HDE it consists of a lot of purine bases right here. So these are the three components that are going to be required for the entire three-step process of the maturation of the histone mRNA. Most of these components come into picture especially in the S phase of the cell cycle. We know this is true because due to the fact that if the description of the S phase of the cell cycle is basically the formation of extra DNA that is to be used by the cell on cell division. So in order to produce more DNA so that DNA can be used by the daughter cells whenever there is a cell division, there is a tremendous increase of the quantity of these components, especially in the early S phase, at about 35 times more than the normal concentration. And then it eventually decreases in the late S phase. Now, let us look at this histone mRNA protein right here. mRNA protein, the histone mRNA protein, the gene that codes for the immature mRNA, does not really have a poly A tail. So according to this information, the next step would be formation of this tail, or the formation of the 3' UTR region. 3' right here. Sorry, 3' right here. Should be 5' here. So the formation of the 3' UTR is initiated is initiated by one of the component that we saw earlier. This component is also known as the SLBP, or the stem loop binding protein. The name stands true due to the fact that this entire protein is binding on a stem loop-like structure. So this is the stem and this is the loop. And this exact same thing is present here, and the stem loop binding protein is binding to it. The next step in this entire process is the recruiting of another common protein that we saw earlier, known as the HDE. The HDE with its rich purine base. Then we have the last component that we saw. That's the U7SN protein. The U7SN protein and HDE are connected together with the help of hydrogen bonds. But they are further connected approximately they're further connected more, especially at the 5' upstream region with the help of the 5' upstream region right there, with the help of a sequence that goes as in AAGAG for the HDE, and the connection by the U7 is done by this sequence, CUUUU. So that's the binding at the upstream region. But that's not it. Then, adjacent to this entire thing, we have something known as the SM binder. The SM binder binds approximately a set of 7 proteins in a configuration of 5 plus 2. 5 being common or the conserved protein that are present in most of the snRNAs or small nuclear RNAs, and the other 2, namely LSM10 and LSM11 that are present right here. So LSM10 and LSM11. I'm going to summarize this more properly. So the internal binding, there is a binding by an SM protein that binds a set of 7 protein inside the U7 snRNA. There are 7 proteins out of which 5 are conserved, that is, they are common across a wide array of snRNA, and 2 are just specific to snRNA, which are the LSM10 and LSM11. Then lastly, we have the last component, which is new, known as the zinc finger. The zinc finger protein 100 to be more precise. Now the main job of zinc finger protein 100 is to bind the components of all the components together. It attaches to the stem loop binding protein, then attaches itself to the LSM10 and LSM10 especially. And then it lastly binds also to the HDE. So it's like a binder. The next process involves the freeing of these components. The freeing of these components is associated with the movement of the U7 protein. The U7 protein moves across the mRNA and then it selects and marks uracil to be cut by endonucleases at sites. So this is one site and then this is another, which leads to the formation of a new type of histone, which only contains the stem loop binding protein and the stem loop. So that's the new mature mRNA. Now that this is formed, the second step involves the translation, the translation by the eukaryotic initiation factor 4E. Eukaryotic initiation factor 4E transports the entire thing in the entire complex into the cytoplasm. So this yellowish structure right here, the background suggests that we are in the cytoplasm now. After that, there's translation. Of course, there might be translation here. There is a removal of the stem loop binder. So the stem loop binding protein gets removed because now the job is done. The next thing that it does is that it brings in exonucleases. Now, exonucleases are destructive in nature. So their main job is to go and bam, just go inside and destroy the entire structure altogether. And that's how there is the formation of the 3' UTR, and then there's translation, and then there's a destruction of the mRNA protein.